In this PPT, we will be seeing about organizer and its types and the experiments done about it. This diagram clearly shows about the Spearman's organization, which results in primary embryonic induction. And let's see in detail about every concept of Spearman's organization. Initially, it is primary induction. It must be understood in terms of movements of cells. This diagram clearly indicates about the primary induction. Mesodermal cells migrate up wall of blastocele. They become arranged as loose sheet immediately under the region of ectoderm, then later becomes neural plate. Dorsal mesoderm induces local ectoderm from neural plate. Here are some experiments about the organizer. Initially, the first diagram shows about the organizer transplant from early gastrula, and it results in additional anterior axis. The second diagram shows the organizer transplant from late gastrula, which results in the formation of tail structures. The next diagram shows about the nervous system is induced during gastrulation. The early gastrulas are taken and which result in the formation of neural tissue and epidermis in the first diagram. The second diagram shows the fragments of presumptive epidermis transplanted from one gastrula to another, and which results in induction of neural tissue and transplanted fragment. The orange line indicates the neural tissue. Induction is region specific, which means that it only occurs in a specific region. The first diagram shows the nerula anterior mesoderm grafted into early gastrula, which induces a head with eyes and forebrain. The second diagram shows the nerula, which is in posterior mesoderm, that is grafted into gastrula, which induces a trunk and tail. You know what the molecular name of inducer is? It is the organizer. Induction is a process which is formed by the result of organizer, by which one group of cell produces a signal that determines the fate of the second group of cell. This implies both the capacity to produce a signal by the inducing cells and the competence of the responding cells to receive and interpret the signal via a signal transduction pathway. Amphibians are the most extensively studied vertebrates for investigations into embryonic induction. For example, amphibian embryo, the dorsal ectodermal cells in a mid-longitudinal region forms a neural plate. The mesodermal layers form the roof of archentron by invagination, which finally forms gut. The diagram in which cells A and B together are induced by an inductor or organizer undergoes differentiation and forms an organ. Without inducer, there is no organ formation. This diagram clearly shows about the external views from side and transverse sections in which an inductor or an organization works. Components of induction. Organizer or inductor, it is the first and major component. These are structures which induce the formation of the another structure that is called inductor or organizer. Evocator, the chemical substance that is emitted by an inductor is called evocator. Responsive tissue, the tissue on which inductor or evocator acts is called responsive tissue. The diagram clearly explains the above mentioned terms. Mangold in 1927, he says the organization of a secondary axis by dorsal blastopore lip tissue. Dorsal lip tissue from an early gastrula is transplanted into another early gastrula in the region that normally becomes ventral epidermis. The donor tissue invaginates and forms a second archentron and then a second embryonic axis. Both donor and host tissues are seen in neural tube, notochord, and somates. Eventually, a second embryo forms that is joined to the host. Structure of dorsal blastopore lip region in an early Xenophis gastrula. One of the embryos was regular and another was induced one. Thank you.